to the chromosomes. Here's a diagram of one of these duplicated chromosomes. So there's the centromere, that's holding the two chromatids together. There's a special protein right around the centromere attached to the chromatids right there and right there, and those are called kinetochores. And the kinetochores are essentially the sockets where those spindle fibers are gonna click into. And so notice that each chromatid has its own kinetochore. The top chromatid and the bottom chromatid each have their own kinetochore. Because what we're going to see here in a moment is that the spindle fiber is attached to both the top and the bottom of each duplicated chromosome. All right, here come the spindle fibers from the centrosome at the top and the centrosome at the bottom of the cell. They sort of reach downward, click into the two kinetochores of each chromosome. So each duplicated chromosome is connected to the top centrosome and the bottom centrosome now. There we go. Well, actually I didn't draw it very well, but this one is connected to the top and the bottom, and the red one also is connected to the top and the bottom centrosomes.
So those centrosomes are the ones that create spindle?
Yes, yeah, they're right. Those are the anchors for the spindle fibers.
Those are located outside of the nucleus?
Yes. Well, remember the nucleus is mostly going to be gone. By the end of prophase, the nucleus is totally gone. But yeah, they are located outside the nucleus. Okay, so there's the prophase. What happens next is that the kinetochores on each chromosome end up sort of pulling on the spindle fibers that are attached to them. And they pull essentially equally towards the upper spindle fibers and the lower spindle fibers, and that results with the chromosomes being maneuvered to the very middle of the cell, which some people call the equator of the cell. Let's watch that happening. So the cell goes into M phase, and notice that the chromosomes are right there in the equator of the cell, right smack dab in the middle between the two poles. As I said, because they're pulling equally hard on both spindle fibers that are attached to them. The nucleus is completely gone at this point. Kinetochore is pulling to the middle. Okay, and you can remember metaphase because the chromosomes are in the middle. It's an alliteration of All right, and now let's go to anaphase. In anaphase, watch the chromosomes. That's when the centromere is split. In anaphase, each centromere broke in two. And so we no longer had duplicated chromosomes. We now have unduplicated chromosomes, half of which are moving up towards this pole of the cell, and half of which are moving down towards this pole of the cell. And so technically they're in anaphase that the number of chromosomes in the cell doubles. We went from, if this was a human cell, we'd go from 46 duplicated chromosomes to 92 unduplicated chromosomes. And let's see a close-up of that. So there's each duplicated chromosome with the spindle fibers attaching to its two kinetochores. The proteins that form the kinetochores, proteins like here and here, not only latch on to the spindle fibers, but they can also pull on the spindle fibers. Maybe an analogy was if there was a rope hanging from the ceiling and I was a kinetochore, I might grab onto that rope and pull myself up towards the ceiling on it. That's what that kinetochore is going to do, and that's what this kinetochore is going to do. It's going to pull itself and the chromatid that it's attached to downward along this spindle fiber like there, and that provides the pulling force to split the centromere and move the two chromatids in opposite directions. And so here they go. And the kinetochores actually chew away at the spindle fibers as they pull themselves along the spindle fibers. All right, so in anaphase, where's our thing? Here it is. Nuclear membrane is still gone. The centromeres have divided, and the unduplicated chromosomes are now moving towards opposite poles because of the kinetochores are pulling them along the spindle fibers. And last but not least, telophase. As anaphase progresses, the unduplicated chromosomes are moving further and further apart towards opposite poles. In telophase is essentially the opposite of prophase. In telophase, the chromosomes are uncondensing. In other words, they're starting to come off the histone proteins and go back into the chromatin state. The nuclear membranes are reforming. A new nuclear membrane is forming at the bottom pole of the cell and at the top pole of the cell. And the spindle fibers are disappearing. The centrosomes and those spindle fiber microtubules are starting to, I guess, get depolymerized. They're just kind of vanishing from the cell. All right, and so telophase is really the last step in nuclear division. Notice that we have gone from one nucleus at the beginning to now two nuclei at the top, one at the top pole and one at the bottom pole of the cell. And after, with telophase being the end of mitosis nuclear division, the next step is, of course, cytokinesis, where the membrane and the cytoplasm split. But to be honest, there's a little bit of overlap between these. That is, while telophase is going on, cytokinesis is beginning. As a matter of fact, you can even see the very beginning of cytokinesis there, where the cleavage furrow is starting to form between the two daughter cells. And there it is, and now we have two daughter cells, each with one set of unduplicated chromosomes. And it was so much fun. Let's look at it again. So what I'm going to show you now, both daughter cells do, but G1 is the first growth phase. More organelles are being added, the cytoplasm is getting larger, nuclear membrane is getting larger. You can't see the chromosomes because there are chromatins all throughout interphase. But if you could, they would be unduplicated chromosomes. And then we get to, yeah, there's what they look like in G1. Well, if you can see them. Then in S phase, even though you can't really see them, that's when they go to duplicated chromosomes. Good, but they're really chromatin. And then G2, another growth phase. And at the end of G2, the cell is big enough and its chromosomes are duplicated, so it's ready to start nuclear division. And that will begin with prophase. I think we're going to get rid of the organelles from the diagram. There we go. Okay, so prophase. The chromosomes start to condense, nuclear membrane starts to break down, spindle apparatus starts to form. Metaphase, the chromosomes get maneuvered to the middle of the cell. In anaphase, the centromeres split. So the number of chromosomes inside the cell doubles at that moment. Telophase, the nuclear membranes start to reform, the chromosomes start to go back to the chromatin. Spindle apparatus goes away and cytokinesis starts. And there's cytokinesis, and we get our two daughter cells. And then each daughter cell would then continue on with the cell cycle and go through the whole thing again. Any burning questions up to this point? All right, well, let's look at some photographs of cells in the various stages of mitosis, the prophase, metaphase, anaphase, and telophase. There's the cell in the very beginning of prophase. You can see that some of the DNA is still condensing. In other words, this part looks like chromatin, but you can definitely see some of the chromosomes have mostly condensed, like there and there and there. And if you look at the cytoplasm, you can see a slight graininess to it there and there. That's probably the beginning of the spindle fibers starting to form. So there's probably some centrioles here with some spindle fibers sticking out there, and it looks like maybe right here you can start to see sort of a graininess coming out in a starburst pattern. Those are also from the spindle fibers. You can't really see the centrioles, I should say the centrosomes very well in these photographs, but they are there. Here's a little bit later on in prophase. The chromosomes are pretty much fully condensed at this point. Here is one centrosome, here's the other centrosome, and you can definitely see the spindle fibers coming out of each one. Here's metaphase, where chromosomes are lined up in the middle, the equator of the cell. And here's anaphase. I probably should have this overhead on. Here's anaphase, the centromeres divide, and so the unduplicated chromosomes start moving towards opposite poles of the cell. And here's telophase. They've separated from each other. The cytokinesis is beginning. You can see this cleavage furrow starting to form right there. And with a little imagination, you can see this one is starting to go back into the chromatin state. This one maybe hasn't quite started yet, but it will soon. And there's cytokinesis, and this one looks even more like chromatin as it's starting to go back into that state. And this one will also soon be going back into chromatin. The next series are some plant cells, but it's essentially the same thing, but except remember, plant cells don't have a cleavage furrow because they have that cell wall, so you get something called the cell plate to divide the two daughter cells. Plant cell in prophase, chromosomes have condensed. Looks like a centrosome there and a centrosome there. Metaphase, the chromosomes are in the middle. Oop, go back. Anaphase, the chromosomes have split apart, moving towards opposite poles. And in telophase, well, they're condensing again. And you can see the very beginning of the cell plate forming right there. That little line right there will eventually be a new membrane and a new cell wall dividing the two daughter cells. Here's a fun slide. This is a slide of some onion cells undergoing, some of which are well, they're all dividing, but you can see them in various phases of mitosis. So just as a review, let's point towards some of these and you tell me what phase they're in. What phase is this one in? It's probably anaphase. Yeah, that's the way it looks like to me. And probably this one is anaphase also. What about this guy? I would say metaphase, because they look like they're mostly in the middle. I suppose it could be prophase, but it looks more like metaphase to me. How about this guy right here? Probably telophase, because they look pretty well separated. I can't actually see the cell plate between the two daughter cells yet, but it's probably starting to form. How about, just for kicks, how about this one right here? What do you think? I think it's prophase, because it looks like the chromosomes have just condensed. The nucleus, you can still see the more or less round shape of nucleus. So the nuclear membrane is probably going away, and the chromosomes are in the middle of condensing. What about this one right here? What phase do you think that's in? Well, I was going to say interphase, because I don't really see any chromosomes in there. It looks very much like chromatin. And so that one's probably still an interphase. Matter of fact, a lot of these where you don't really see any thread-like structures are going to be an interphase. That was interphase, that was interphase. How about this one right here? Cytokinesis. It looks like the two nuclei have formed. There's the cell plate right there, and they're going back into the chromatin. Sounds like my time is up. Okay, I will see you guys in laboratory today.